All right, just as a reminder, you know that you have a lab test that you've had a review for since like week 3. I had a few questions this morning and I was like, guys, if it's not in the review, it is not in the test, meaning that for test 1, we stopped at the endospore stain. Remember what happened was that we had to shift the endospore stain to the following week. So that kind of made it so that we had material that was part of test 1 for the lab at the same time as we were covering the new material for test 2. So just think about that. So if it's not in the review, in the lab review, it's because it's part of the next test. So the phenol red, the gelatinase, for example, all of that was part of the next unit in lab. Now, I had some questions this morning about the test, and I'm like, well, guys, you know, the syllabus tells you that you get 1 minute per question. That's not different. You probably have seen jokes that professors want to wear t-shirts that say, read the syllabus. Well, we write the syllabus because those are basically the rules that we're supposed to abide by. So, if you look at the syllabus, the description for the lab test include things like multiple choice, short answer, fill in the blanks for the most part, and every now and then you may see a true false. So that is the type of question that you get in any lab test. It doesn't mean that you will always have every single type of question, but that is the question type that you'll see. You still get 1 minute per question, but what I've done is that I've given you 22 questions because it's supposed to be 44 points per the syllabus. And I just gave you 25 minutes in case you needed a few minutes to wrap things up, a few extra minutes to wrap things up. By the way, this is the same way we do it in face-to-face exams, except that they don't get 25 minutes, they get 22 minutes. Remember that we are supposed to have time tests for you. Our health professions department asked us that we have time tests because they provide a lot of time tests themselves. So you're going to log into Respondus for the lab test, and it is a time test. Are there any questions about the lab test before we begin with the lab for today? So if you're asking if you can backtrack, the answer is yes, but do it carefully. I used to not let students backtrack, and there's a reason for this. When you are backtracking, even if you do not use Respondus, students sometimes press, let's just say that they get a little anxious or nervous or just not patient enough, whatever it is, and they try to backtrack multiple times at the same time, basically within the same second. It can make it so that you accidentally submit your exam early, which is why for a while I stopped that feature. But students told me that they like backtracking, so I put it back. So I think that's what you were asking, Shady. If you could backtrack. The answer is yes, but be very careful when you're doing it. I get very nervous about backtracking because I know what can happen. And if you're wondering, how do I know? So I had students panic that accidentally submitted their test too early. How do I know that that is what happened, by the way? There's a log in Blackboard and in Respondus that tells me how many minutes you actually spend in each question. And I can tell if you're actually telling me the truth or not, by the way, because students have accidentally submitted their test in minute number 2 of a test that had a total of 25 minutes for doing it. And I know that in 2 minutes, you cannot go through the whole test. So that's how I know that they have accidentally backtracked. So if you want to do it carefully. Be patient. Let it load. But what you need to take into consideration is that that may take some time, a few extra seconds. It's not going to take forever, by the way. It depends, of course, on your connection. If your internet connection is poor, then backtracking is going to take forever. I'm not going to lie. That's just the way it works. Stuff has to load. Are there any other questions? And now my dog has decided to drink water right next to my computer. There's 3 bowls, but he decided that that's the one. So I apologize if you hear that. He has decided that you're going to listen to him today, either snoring or drinking. All right, so let's go ahead start it with the new labs for today. And remember that today is when we're going to cover the last things that you have to do for your unknown. Now, I had a student this morning asked me why we were doing new labs if we had a lab test. And I'm like, well, in real life, we probably would have done it this way. But really, I wanted you to have extra days to work on your unknown. Because if we do everything on Wednesday and you have your work due on Friday, you may need some extra time. So just keep that in mind as well. Now, we are going to go over some differential testing today. But something that I started saying last time we met is that always try to think of why or when you will go through the trouble of setting up a test in the laboratory. For example, what we're going to go over today is something that we call the IMVIC series. And in the IMVIC series, you're going to inoculate different types of media to get different results that are part of what we call the IMVIC results. Now, that probably makes you think already that this has to be extra work because you are doing several tests in one series, in one sitting. You're doing several inoculations and incubations in one shot. So, when will you go through this trouble? If you are in the process of telling apart a member of what we call the Enterobacteriaceae or enteric bacteria. What comes to mind when you think of enteric? What does enteric remind you of, that word? Intestines? Sure. Yeah. And I mean, and if you thought poop, yes, that's true. So enteric bacteria are bacteria that tend to inhabit the intestinal tract. Now, what we see is that many of the members of the enteric bacteria are gram-negative bacilli that ferment lactose. So if you are inoculating an IMVIC series when you're identifying bacteria in the lab, you already did a gram stain that told you that this was a gram-negative bacillus, and you tested for glucose fermentation. What test could you have done that we learned about not too long ago that would have given you this result for lactose fermentation. Yes, the phenol red broth, yes. Because in the phenol red broth, we could test for lactose fermentation or glucose fermentation or sucrose fermentation or even other sugars. So if you did a gram stain of an unknown in the lab, and it was a gram-negative bacillus that fermented lactose in the tube, it turned it yellow, then you will think, I wonder if it is an enteric bacteria, because many of the enteric bacteria are lactose fermenters. For example, Shigella can colonize the intestine, that's why it's enteric. E. coli, Salmonella, those are the most common ones. Yersinia is one that can colonize the intestine. We don't see it as often, but sometimes we do see it. Then we have here Klebsiella pneumonia, which can inhabit the intestines, but the name tells you that it can also live in the lung tissue. It's one of those that's very interesting. Now, there are a few others that we don't see very often as far as infections of the intestine, like Providencia, Morganella, and Enterobacter. All of those are lactose fermenters. And it would be a good idea to set up that INVIC series so that we can start separating them. So what we're going to do is go over each of the media that are part of the INVIC series. So first of all, INVIC stands for indole methyl red. The M is for methyl red. The V is for the Voges proscour, and the C is for citrate. That is 4 different results. But here's the thing, and this is very interesting. That indole test, the I in the IMIC, doesn't just give you the indole result. It's a media recipe that we call SIM. S for sulfur reduction, I for indole production, and M for motility. So technically, you can get 2 extra test results here. They're not necessarily part of the INVIC series, but you can get also motility and sulfur reduction as part of the deal at the same time. Are there any questions so far? All right, we can get started with that one with the SIM. So they're dips. So it's media that's solid in a tube. It's not slanted. That's why it's a dip. And after the inoculation, you will be able to read 3 results. And one of those results happens to be the one that's part of the INVIC series, which is the indole production test. Now notice that at the tubes, the picture with the multiple tubes at the top, there's all sorts of colors and variations. Still the same media, it's just different bacteria that's growing in it because they have different results for the sulfite, for the indole, for the motility. So let's get started with the S portion, which is the sulfite reduction. First of all, it is a dip, so you inoculate with a needle, like when we talked about that motility test previously. After incubation, you're gonna look for different things like sulfur reduction. Now, these 2 tubes are inoculated. meaning that bacteria is growing in them. Now we are going to refer to the three results independently from one another, but know that you will always read the three results for each of these tubes that you inoculate and incubate in the lab. So the results are going to be either production of a black precipitate, as you can see in this tube, or no precipitate. The interpretation of results, if there is no precipitate produced, then will be H2S negative or no sulfur reduction. For the tube that shows a black precipitate as a result, the interpretation of result will be sulfur reduction or H2S positive, because you have that black color that indicates H2S in the media. Are there any questions about the sulfur reduction? All right. So let's look at the M portion of SIM, which stands for motility. Now, if all you care about is to learn if an organism is motile or non-motile, you could do the motility test that we learned about in Unit 1 of the lab, or you could do one of these SIM tests. Now, I always think that the motility test tubes are easier to read than these SIM tubes when it comes to motility, especially for students because they may be seeing this for the first time in their lives. And in the motility test, we had a color indicator that made the growth look red. And you can see here that the growth does not look red because there's no color indicator. Now, because there is no color indicator, what we do is that we hold the tubes against the light, or maybe even sunlight through window, and compare them with an uninoculated tube. If you see a result of a straight line like this one, that can be interpreted as an organism that is not motile. If you see a result where there is growth in the line, but there's also diffuse growth throughout, that can be interpreted as that organism being motile. Now, before we move on, I gotta point out something. In your lab manual, if you are using the lab manual, it tells you that if you see sulfur reduction throughout the tube, like this one, that you cannot determine motility using the SIM because you cannot really see the growth. Now here's the thing, and this is true. The reason why there is sulfur reduction throughout the tube is because there's bacteria everywhere. It's because it's motile. If it was not black throughout, because the bacteria is not motile, the black line is going to be confined to being around the growth instead of everywhere in the tube. But if you're reading your lab manual, basically they're playing it safe, saying that if there's black precipitate throughout the entire tube, that you cannot determine motility. Are there any questions about the motility portion or even that sulfur reduction portion? All right, now we're gonna go to the I in SIM, and I left it to the end because this is the I that starts the INVIC series. So I for INVIC, I for indole in INVIC. So the way it works out is that in order to read the indole production result, you're gonna have to add a chemical. Now, let's talk about where this indole is coming from. And I have to backtrack to a picture I used earlier here, where you have these molecules at the bottom. If the bacteria has the ability to produce an enzyme called tryptophanase, I'll write it over here. Then that bacteria is going to be able to break down the amino acid tryptophan that happens to be part of the recipe of SIM. And what is produced in the end is something called indole. Here you have tryptophan. Notice that what's happening is that this portion of the molecule is removed. So we add a reagent to the top of the tube. We add 6 to 8 drops of Kovacs reagent. And you can see in the bottle here is yellow to start. And what we're going to be looking for within seconds, really, you will be able to see a color change or no color change. So in these two tubes that I have here, here is the Kovacs and here is the Kovacs reagent. There is no change in color in the tube on the left. There is a change in color to a pink-red color on the tube on the right. That can be interpreted as, for the pink-red tube, there was indole production, or it is indole positive or tryptophanase positive. On the left tube that has no change in color, because the Kovacs actually shows you the presence of indole, it reacts with it. There will be no indole production or indole negative or tryptophanase negative. Are there any questions about that? All right. Now, we just did the I portion of INVIC. We're going to move on to the methyl red and the Budges-Proskauer next. And we're going to start by saying that for both tests, you're using the exact same media. It's something that we call MRBP media. It's broth. And there is glucose as part of the recipe of the media. And depending on the metabolic processes, the enzymes that the bacteria have available, they may or may not be able to use glucose, but also those that use it may do so differently. And that's why we actually run two tests out of the same media. What we do is that we inoculate the MRBP broth, like this one that you have in the picture, which is kind of like a light yellow color, with a loop. We incubate that for 24 to 48 hours. And then we split them up into two clean tubes. To one tube, you add the MR reagents. To the other tube, you add the VB reagents. Now, I'm not going to lie to you, that can be messy. You can spill bacteria. So to prevent you from doing that, sometimes I just have students inoculate 2 MRBP tubes so that you can do the MR test in one tube for that bacteria, and in a second tube, the BP test. So for MR, what you're going to be able to detect is whether or not glucose was fermented and there was production of acid. Because methyl red is a reagent that is going to act as a pH indicator. So after you have inoculated and incubated for 24 to 48 hours, you add methyl red to the tube and you tap mix it to make sure that all of the methyl red mixes in with the liquid. And you're going to look for a color change as a result. By the way, this tube looks really red here. Usually it's more like a pink. I'm not going to lie to you. In real life, it's going to be more like a pink. And here you have no change. Now, let's talk about what that means. Let's talk about how we interpret that change in color. If there is that pink or reddish color, you are going to say that there was acid produced when the glucose got broken down. That would be also MR positive. If there's no change, then no acid was detected from glucose breakdown, and that would be MR negative. Now, when we look at the Voges-Pro scour test, what we're going to detect here is a little different. We're not looking for fermentation per se. We're looking at the use of glucose in a different type of process. So the way it works out is that what gets produced in this case, instead of an acidic byproduct or acidic waste, when glucose gets used up, is something called acetoin. And this acetoin can be detected by adding reagents. The same way the acid could be detected by the MR when we added methyl red. It's just that we're going to use different chemicals this time. So after the incubation, what we do is that we add 12 drops of 30% potassium hydroxide, which usually we label in the lab as BPA. And by the way, if you know that you have a hard time remembering potassium hydroxide as a word, BPA is just fine. And we also add 4 to 5 drops of alpha naphthol that in the lab we also refer to as BPB. Now you're going to tap mix this. And if you read the lab manual, it tells you that you're going to tap mix those tubes or tube every 10 minutes. Yes, for 1 hour. Because it can take that long for the chemical reaction to take place to detect that acetoin. What we do, we're kind of sneaky, is that we add something else in the mix. We add a powder called creatinine that is going to make it so that the reaction happens faster. It's going to be a catalyst of sorts. And we mix by tapping, and usually you cut the time in half to see the color change. It may be 10 minutes, by the way. Usually 10 minutes is enough. Sometimes we have to go up to 30. So then you tap mix every 10 minutes for half an hour if you add the creatinine. And you're going to look again for a color change. That's all you care about. Now, when you add the BPA and the BPB and the creatinine, you're going to have a bronze color. But that's not what you're looking for as a positive, by the way. And if you add them, sometimes you see this red color. These colors have different meaning. If it's bronze, there was no acetone production. It is BP negative. If it is red, though, that's the color you're looking for to indicate that acetone was detected and that that organism is BP positive. Are there any questions so far? That's not me, it's the dog. Sorry, he was nudging me to pick him up, so sorry. I figured that will give you time to ask me a question. If not, we'll just keep going, and I'll just keep holding the 20 pounds worth of dog. All right, now, at the end of that IMBIC, there's that C. I feel like I'm doing Sesame Street. C is for citrate. Yes, it is. Or citric acid. So what we have here is just we're looking for the breakdown, or no breakdown really, of this citric acid. So obviously this has to be something that happens because of an enzyme. There's different names for this enzyme. The one that's most commonly used is citrate permease. Some books call it citrase. That's the shorter version, I guess. Now, the way it works out is that this media offers citrate as a carbon source. And what happens when citrate gets used up is that there's going to be a change in pH to a more alkaline or basic pH so that it gets darker. So it goes from this beautiful green color to something like a deep blue. Now, when you inoculate this, you can do it with a needle. You stab the bud and then you streak the surface. Sometimes you can just stab the butt of the slant with the needle, and you can use a loop to add bacteria to the slant. Then you will incubate this and look for that color change or no color change as a result. So if you see a green color, which is what you started with, that will be interpreted as no citrate permease produced, citrate negative. If you see that beautiful blue color that you see on the left-hand side tube, that will be production of citrate permease or citrate negative. I'm sorry, positive. And that will be the end for that IMVIC series. Now, depending on the combination of results that you have, maybe you have positive, positive, negative, positive, or different combinations of these plus and minus signs, you're going to be able to narrow down to what possible enteric bacteria that was fermenting lactose earlier this can be. Are there any questions so far? Okay. So you know that we're going to go on to our unknown next. What you have to do. Literally, I just took screenshots of my Blackboard because many of you have trouble finding some of the items in Blackboard still. And I know that that week and a half off was a mess. It's almost like we got to start from scratch. So you're gonna open the lab folder for week 7, and in there you have 2 folders, one for Monday, which is today, and one for Wednesday. And in the Monday one, you'll see a folder that says Finish Morphological Unknown, and you have some instructions there. I always put them multiple places so you can't miss them. When you open that folder, the first thing that you're gonna do is find your unknown number and read the last set of results. It may be an endospore stain if your organism was a gram-positive bacilli. It may be a catalase test if your organism was a gram-positive cocci. Or it may be a motility test if your organism was a gram-negative bacillus. After you have read your results, you're going to scroll down. There's a couple of documents that you're going to have to look at. The first one is labeled Unknown Table with all possible unknown result tests. And when you open it, it looks something like this. These are the exact same bacteria that you already work with in exercise 2.2 for colony morphology on week 3 of the semester. It's just that all you have for them is that colony morphology description. And that is not going to be enough for you to tell me what bacteria you have. So here you have to use this time around the gram staining result, the cellular morphology and arrangement. This is something different from colony morphology. The motility result, the catalase result, the endospore result. You will ignore this acid-fast stain because we didn't cover acid-fast this semester. Now, this looks a lot like a table that you could potentially use to create a dichotomous ski, like you did in your homework one. Now, I know that it's been a while since you have created a dichotomous ski. Because of that, I once more put the links to the videos that help you understand how to construct a dichotomous ski. For example, there's that one that uses actually bacterial results in the lab. But what you're gonna do for Friday is going to be to open that document that's labeled Final Unknown Submission. And you will write down your unknown number. You will type down what you think the name of your unknown is, the genus and species. The last set of results that you read, which were either catalase or motility or endospore, and the interpretation of those results. And you're going to paste in there a dichotomous ski, just like the dichotomous ski that you created for the homework. It's just that you're using different tests this time around to separate the bacteria. And this is due on Friday by noon. Are there any questions about the unknown? So this is the last piece of information that I have for you for lab today. So are there any questions before we adjourn for today?
I have a quick question. So on the table that you're gonna do the dichotomous key on, we're gonna put each of those tabs, correct? like the gram, the motility.
So that you're asking if you have to use them all to be able to separate them to get to your answer, because what you want to do is get to your answer. So yeah, you will end up using them. Yes. Now, something that is not on the table, Shady, that you have already because you made it yourselves, is that colony morphology table description. Because at the end of it all, you may be between two bacteria, and you're going to have to, maybe, not for all of you, but maybe go to the colony morphology plate description that you did at the beginning of all of this, and look at that table that you created for exercise to do, and think which of these two bacteria may be based on that colony morphology. That's what we call the morphological unknown.
Okay, thank you.
You are welcome. Anything else? All right, remember that you have until 11:59 p.m. to take your lab test. And what do you have due tomorrow? If you haven't done so already. Yes, Eduardo, because that is your unknown. You cannot mix and match numbers because that is your unknown. Now, no one answered. What do you have due by tomorrow? Yes, the lecture test. I know that it seems like I'm micromanaging you, but man, something happened during the storm, and well, the connections have to be rebuilt. And I don't want anything to fall through the cracks. All right. Have a good rest of your day. And please stay warm. These weather changes are going to make everyone at some point just, your metabolism is going to go crazy. One day you're hot, the next day you are cold. All right. Have a good day, everyone. Thank you so much for joining me. Happy Monday. Today is National Pig Day of all the days. It just occurred to me that it is. All right. See you on Wednesday. Yes, National Pig Day.